Hi, I'm Gabby. I'm from the Venuscular group in the chemistry department, and today I'm going to be talking to you about drug targeting of intrinsically disordered proteins. Before I begin, I'd first like to introduce what a protein is. Proteins are biological machines made up of amino acids, and the sequence of these amino acids corresponds sometimes to a very rigid 3D structure of the protein. And this structure is very correlated with the protein's function. Proteins are very responsible for all sorts of biological processes in cells in our bodies, including the transport of oxygen, the ability to see, and inflammation. In fact, proteins are so important for all biological processes that nearly all pharmaceutical drugs directly target proteins. And the way that they target them is through the lock and key binding mechanism. And this lock and key binding mechanism depends very much on the rigidity of the protein target. And the way that it works is a drug molecule can fit directly into the active site, or the site where relevant chemistry happens within the protein, like a key fitting into a lock, and directly affect that protein's function. But when you're developing drugs to target active sites, it's really important to really understand the rigidity of the active site. Now, unfortunately, not all proteins are well structured, including the proteins that I study. For example, this is one structure of a single protein that I study, but this protein can also be represented by all of these other structures. And my protein is rapidly interconverting between these structures. So as you can see, there's not really a single active site that I can target, despite the fact that this protein is very relevant in disease. These proteins fit into a class of proteins called intrinsically disordered proteins. As I mentioned, these proteins are highly dynamic, and because they're so dynamic, they're very difficult to study experimentally. Furthermore, they're highly prevalent in disease. Over 50% of cancer-related proteins are considered disordered proteins, and these proteins are also very, very associated with neurodegenerative diseases, such as Alzheimer's disease, Parkinson's. These proteins tend to form these plaques, which interfere with biological processes. Because they're so dynamic and so difficult to study experimentally, these have been deemed untargetable by drug companies and drug developers. But my group is now developing a new mechanism to target these proteins and alter their behavior, which we call entropy-driven binding. So what is entropy? Many people think of entropy as disorder of a system. That's a little abstract. I prefer to think about it in terms of counting states. So for example, consider the following room. It's very clean, and everything has a very specific place. So there's really only one way for everything to be in its proper place. So we say the number of states or ways that this room can be tidy is 1. But what about when the room is messy? How many ways are there for this room to be messy? Well, there are many, many ways that this room can be messy. Here is one example. But alternatively, the chair could be on the other side, more books could be on the floor. And so we say that there are many, many states in which this room could be messy or disordered. So this can give us some intuition about entropy. We would say that the clean room has a very low entropy, but because the messy room has many, many ways in which it can be disordered, we say that it has a very, very high entropy. How can we use this to alter the behavior of disordered proteins? Well, as I mentioned, my proteins exist in many, many states. So we could say that they have a very high entropy as compared to the other more structured proteins that are typically targeted with drugs. And I mentioned before that these proteins are you're commonly associated with disease. But interestingly, not all of the states of my protein are associated with disease. Rather, only some of them, such as the 4 that I've highlighted in a red circle. So what I aim to do is develop drugs that increase the total number of states of my protein. So if we can introduce more states or make it more disordered, we can sort of have disease prevention by numbers, rather diluting the pathological states of our protein. So how do we do this? How do I study it? As the organizers mentioned, this is a small session. We study small things. So you can't actually see a protein under a microscope. We have to model them on a computer. And I use an approach called molecular dynamic simulations. And the way that it works is I model every atom within my protein and every atom within the water molecule surrounding it, and every atom in my drug in a computer. And I integrate Newton's equations of motions using supercomputers to understand the behavior of my protein over time and how it moves and binds the drug. The problem with this is, depending on some of the parameters we use, we can get different results if we slightly change them. So we have to use experimental techniques to restrain our simulations to make sure that what we're seeing on the computer is consistent with what we see in the test tube. So what do our results look like? So we actually are able to generate videos of our protein binding. You can see it's very highly dynamic. both the protein and the drug, and the drug doesn't always bind in the same spot to the protein. In fact, throughout the entire simulation, if we overlay in red where the drug sort of sits, over time we can see that it's really well distributed along the protein backbone or along the body of the protein. Can this be physiologically relevant? So as I mentioned before, Alzheimer's disease is a problem of protein aggregation. And so we've tested whether this drug can actually inhibit protein aggregation. And I don't have time to get into the details now, but just to give you a few results, we have seen that these drugs, via this entropy-driven mechanism, can actually inhibit protein aggregation, which is really promising as a technique for Alzheimer's, Parkinson's, et cetera. With that, I would like to thank my group, the Vendor School, specifically Francesco Aprile, Max Bonomi, and Carlo Camilloni, as well as the Churchill Foundation and Churchill College, also the organizers. Thank you for this opportunity.